in an experiment.
We don't know yet.
Why is light so fast?
Like, it sounds so simple.
They had no idea.
But now the data speaks.
I find this not only refreshing, but at some level astounding.
Nature.
Welcome back to the Nature Podcast. This time, an AI-powered graphene tongue with a discerning sense of taste.
How migrating salmon can cycle both nutrients and contaminants.
And the 2024 Science Nobel winners. I'm Nick Petrichow.
And I'm Benjamin Thompson.
Researchers have made an AI-powered graphene tongue that can identify subtle differences between different kinds of drinks.
We take different Coke products, zero sugar Coke, caffeine-free Coke, because all of them will be very sugary. Unless you drink Coke very regularly, you won't be probably able to tell the subtle difference between a zero sugar Coke and a caffeine-free Coke, right? But the network is able to learn this.
This is Saptarshi Das, one of the researchers behind the Graphene Tongue, a device with a rectangular graphene sensor on a kind of circuit board that can be plugged into a computer for the AI to work and provide analysis of liquids. Fans of the Pepsi challenge will also be happy to know that the tongue device can even tell the difference between Pepsi and Coke, with a high level of accuracy. And whilst this may sound like just a fun party trick, this use of AI to make an artificial tongue that can tell the difference between very similar kinds of drinks is actually showing that it's possible to overcome one of the issues of using graphene as a chemical sensor. Now, researchers have been interested in using everyone's favourite wonder material as a chemical sensor for a long time. Graphene itself is very chemically inert, and so won't react with whatever it's being used to sense. And it can be a very sensitive detector as well. When a chemical is added to graphene, a layer of ions forms over it, which will have subtle variations depending on what the graphene is interacting with. If you can read those variations, you can figure out what the chemical is, potentially opening up the possibility to detect all sorts of things. But getting that to work in practice has been tricky, as graphene has a bit of a reliability problem.
The problem is when you go and make a device, you will see a lot of variation from one device to another device, right? Even when you use the device today versus tomorrow, you will be seeing some difference in the response of the device.
Not ideal if you're wanting to do very sensitive chemical analysis. One strategy to avoid this may be to try and make the device itself as reliable as possible, removing the variations you see day to day. But Saptarshi and his team went for a different approach and looked to biology for inspiration.
If I think about what is the best chemosensor, which is naturally available, right? The first thing that will probably come to your mind is our tongue.
And similarly to graphene-based sensors, our tongues are not exactly the same person to person. And yet, if I was to do a blind taste test of a chocolate cake, and so were you, we'd both be pretty likely to be able to tell that it was indeed a chocolate cake and not, say, a Victoria sponge. Which led Saptarshi and the team to think about what's enabling that. And they landed on the idea that it's not the hardware of the tongue, but the software that's allowing this. When we grow up, we learn the differences between different kinds of food, and we train our own gustatory cortices in our brains. So the team wondered if they could achieve a similar thing with AI.
So the graphene, therefore, we thought about using as an electronic tongue because it can actually sense different chemicals. And then the artificial neural network or different machine learning algorithms serve as a gustatory cortex
So they trained the AI with the information from the Graphene Tongue on what different kinds of drinks are like. As I mentioned at the start, by doing this, it could tell the difference between Coke and Pepsi. But there are also more practical applications. For example, the device could be trained to tell the difference between regular milk and milk that had been watered down. Useful if you wanted to, say, make sure no one is trying to pull a fast one on you when they're selling you milk. And it could also be trained to find out if things have started to go bad.
We kind of put the orange juice with day 1, day 2, day 3, day 4, and then the network is able to also figure out whether it is kind of old or it is freshly squeezed.
This ability could have obvious applications in food monitoring and safety. But Huangjing Lin, a researcher of nanochemical sensors who wasn't associated with this study, was more impressed with the potential for the AI-powered method to overcome the reliability problems that have plagued chemical sensors.
So I think they provide another possibility that even though with this variation of the device itself, we can integrate it with this artificial neural network to realize the enhancement on the reliability. So if this approach can be applied to more biosensors, I think it will be very beneficial to the development of research in this field.
She did wonder though how well the sensor might work if there was something unexpected in the liquid it was sensing.
I'm not sure how the other interference, for example, some other chemical that mix in milk or this Coke or this drink, whether this interference will introduce some other variation in this approach.
Shaptashi says that if something unexpected was in a known drink, the device and AI would be able to determine that something was different. But he acknowledges that the Graphene Tongue is quite specific, so it wouldn't be able to figure out what the unknown thing is. If you haven't trained a neural network on what a specific drink is, it won't be able to identify it.
If I just train it to detect what is a good orange juice versus a bad orange juice, and if I don't train it to determine whether the Coke is zero sugar or caffeine free, it will not be able to perform. So I need to go through that training process, and the training process will be very problem specific.
Still, he believes that it could have a range of applications in the food industry. To get there though, he thinks it'll need to be made maybe a bit more user-friendly.
You have to then think about packaging these devices so that, you can have these handheld so that any person, let's say, for example, today when you're drinking a tea, right, you can literally dip this sensor, and then you can detect whether the tea is what you were actually thinking to drink, right? So therefore, a lot of packaging aspect to make it really more commercial and more like user-friendly.
He thinks that this can be achieved in the next few years. So perhaps we'll see graphene tongues out there in the world sometime soon. In the meantime, Shaptashi, like Huangjing, thinks that this AI method shows a way to make great sensors without having to have the perfect material.
For many problems, it may not be necessary to make the perfect device first and then put it into application. Some of the application can occur even with imperfect devices. So that is kind of the take-home message that I wanted to kind of give to the community through this paper.
That was Shaptashi Das from Penn State in the US. You also heard from Huangjing Lin from Southern University of Science and Technology in China. For more on that story, check out the show notes for some links.
Coming up, new research looking at the levels of nutrients and contaminants that Pacific salmon carry during their epic migration. We'll also be hearing about the winners of this year's Science Nobels. Right now, though, it's time for the research highlights with Dan Fox.
Scientists have designed a 3D printed microscope powerful enough to detect individual molecules. State-of-the-art microscopes are often bulky, expensive, and difficult to use outside of controlled laboratory settings, which can make them impractical for many researchers. To tackle this, a team developed a microscope system that is compact, affordable, and versatile. It combines a 3D printable 4-layer structure with simple optical components such as mirrors and filters, which can be bought from commercial suppliers. Interchangeable layers and optics make this new microscope, called the BRIC MIC, useful for a variety of tasks. The team hopes that this Swiss Army knife microscope system will be used in research labs and beyond. If you want to take a closer look at that research, you can read the paper in Science Advances. Newly hatched sea turtles swim their way out of buried nests into the world. Green sea turtles bury their eggs, meaning hatchlings emerge under up to a metre of sand. So the first challenge these tiny turtles face is digging their way to the surface in total darkness and with limited oxygen. Laboratory observations had suggested that newborn sea turtles randomly thrash their way to the surface over 3 to 7 days. To look at this behaviour in the wild, a team of researchers attached activity sensors to 10 newly hatched green sea turtles. They saw that the newborn turtles first orient themselves to face heads up, possibly guided by gravity, before pitching backwards and forwards to move upwards to the surface. This swimming technique helped the turtles get to fresh air in around 1 to 4 days. Don't bury your head in the sand. Read that paper in full in Proceedings of the Royal Society B.
Next up on the show this week, we've got a story investigating the role that migratory salmon play in cycling nutrients and contaminants between ecosystems. Now, within an ecosystem, a food web is the pattern that explains who eats whom. But while we often consider these webs in isolation, the food web in a wood, say, the boundaries between them can actually be quite fuzzy. Migratory animals, for example, can play a role in linking them together. One group of animals are Pacific salmon, as Jess Brandt from the University of Connecticut in the US explains.
The 5 main species of Pacific salmon, these are the pink, sockeye, chum, coho, and chinook salmon, begin their lives in freshwater systems. They're born in rivers and streams and lakes, and then migrate out to coastal and open ocean habitats where they will do most of their growing. They accumulate something like 95% of their biomass by feeding in marine food webs. Then at the end of their lives, they are prompted to return back to the same freshwater systems that they were born in. That's where they will spawn, and that's where they will die.
With unerring accuracy, these animals somehow find their way to the freshwater spawning grounds they came from. And they return in huge numbers, obviously a lot bigger than when they left. This mass movement of fish also means that many different chemicals they've picked up along the way are also migrating along with them too, which has been of great interest to researchers.
What has been especially well studied is the effects associated with the nutrients that they carry in their tissues. The macronutrients and the micronutrients that they accumulate from marine food webs that they bring back into freshwater food webs.
Because Pacific salmon are of great importance culturally, ecologically, and economically, this aspect of their impact has been investigated for many years. These fish deliver things like carbon, nitrogen, and phosphorus, and important nutrients like omega-3 fatty acids directly into the freshwater food webs. But there's a flip side that hasn't been looked at quite so much.
A lot less research, but some very important research has shown that Pacific salmon bring contaminants back and they release them into those systems through the same processes when they reproduce, when they excrete, when they are consumed or otherwise decompose in the systems. And what's been a key finding of some of that research is that salmon can represent the most important source of contaminants to some of those systems. And that's especially true in systems where there are not direct sources of contaminants to the environment, some remote areas where there's not industrial activity or a lot of agricultural activity, for example.
By and large, these two aspects have been studied in isolation, something Jess and her colleagues wanted to address in their new paper.
We are comparing and contrasting the bright side of subsidies, the potential benefits of these subsidies, and the dark side of those subsidies, the potential harms. This is one of the main ways that our study builds upon the research that's been done in this area. We are considering both nutrients and contaminants together because we think that they have the potential to offer unique insights about how changes in the Pacific salmon community are affecting the influence of Pacific salmon in recipient systems.
And the systems they looked at were spread across the northwest of North America, from salmon migration runs right up in the Bering Sea down to those in California. And they did so over a time span of 40 years, from 1975 to 2015. The team wanted to find out the levels of different nutrients and contaminants, how and where those may have changed, and whether contaminants had reached ecologically significant levels. They looked at 4 types of contaminants, ones that stick around in the environment and gather in animal tissues. These included mercury, industrial contaminants, pesticides, and flame retardants. The team combined existing data from different sources to get an estimate of the amount of each species that returned each year, and the levels of nutrients or contaminants that each fish carried. The team's data suggests that a staggering number of salmon return to their spawning grounds each year.
Over the 40-year time period that we considered, we estimated that the average return of Pacific salmon is 119 million fish at the continental scale. So that involves thousands of tons of nutrients and kilograms of contaminants.
But the quantities and compositions of these changed over time. Evidence has shown that Pacific salmon are getting smaller, associated with competition for food and climate change. But Jess and the team found that this decrease in size was offset by an increase in overall numbers and also biomass of fish.
We saw more than a 30% increase in the biomass of the Pacific salmon. That translates to a 30% increase in the nutrient transfer and a 20% increase in the contaminant transfer. Both nutrients and contaminants increase, but nutrients increase proportionally more than contaminants did.
And there's one type of Pacific salmon that helps explain both this overall increase in biomass and why nutrient levels have increased at a proportionally higher rate. That fish is the pink salmon.
Pink salmon are relatively small organisms. They mature over one winter at sea, and they eat relatively low on the food web.
These smaller, short-lived salmon don't accumulate as many contaminants as those further up the food chain, so they have a higher ratio of nutrients to contaminants than other species. Also, pink salmon populations have been increasing, and they now exist in much higher numbers, which helps explain the shift towards the current overall positive ratio of nutrients to contaminants. But while this was seen at a continental scale, there were regional differences, with ratios changing depending on the levels of each species present. This work provides a picture of the levels of nutrients and contaminants salmon are bringing from the ocean to their freshwater spawning grounds. It's worth noting that while levels of these might represent, well, a drop in the ocean compared to levels pumped out to sea by things like human activity, there is a key difference, as these chemicals are concentrated.
The nutrients and contaminants that salmon include in their tissues, those are packaged as food. They're ready to eat, and they're ready to assimilate into the tissues and the bodies of the animals that eat them. And they're, in the case of contaminants, they're in the chemical form that is the most bioavailable and ready to interact with the biology of the consumer.
So in a sense, the salmon are focusing things into a small area and have already converted them into forms that can be absorbed by other animals. Obviously, for contaminants, this could prove to be a significant issue, even if levels are relatively low, such as with mercury.
The amounts of contaminants that are coming in, those are in the kilogram range. Those numbers surprised us. We were especially surprised to find that only 7 kilograms of mercury were coming back on an annual basis. But what that prompted us to do was to ask the next question: could that amount of mercury be important or significant to the consumers?
Returning salmon are eaten by a lot of types of animal, so the team did some modelling to identify potential harms, using one species as a case study, the bald eagle.
There are about 65,000 bald eagles that span the same spatial range that Pacific salmon return to. And we asked if all of those birds ate an equivalent portion of salmon and assimilated just a fraction of the mercury that is in the salmon tissues, will they achieve concentrations that can be harmful? And we found, yes, even by assimilating a small proportion, the bald eagle population could achieve mercury concentrations that would exceed health thresholds. And that tells us that just a little bit of the contaminant burden that Pacific salmon transport can be toxicologically relevant.
And in terms of toxicological relevance, what might it mean for one particular consumer? Us. There's a lot more research that needs to be done, but Jess's work offers some clues.
We calculated a risk threshold that takes into account both the benefits of the nutrients that you get from eating salmon and the risks that you take on through the contaminant exposure that comes from the fish. All 5 species of fish had scores indicating that they bring a net benefit to people. So the nutrient exposure outweighs the contaminant exposure, if you will.
Further evidence and public health data will need to be collected to quantify exactly what's going on, because the nutrients are not the direct opposite of the contaminants, and their individual specific risks and benefits will have to be calculated, which could be different for different populations. Say, folk buying tinned salmon in a supermarket versus the indigenous peoples who have strong dietary and cultural ties to Pacific salmon. But while Jess's work might not answer these questions, it does provide a new estimate at both continental and local levels of the role these animals play in cycling nutrients and contaminants. Lily Twining, an ecologist from the Swiss Federal Institute of Aquatic Science and Technology, has co-written a News and Views article about the research. For her, it was combining the positive and negative that is this work's key aspect.
So the Pacific salmon system really is like a textbook example of animal migrations moving nutrients. But what I thought is really interesting is that the authors are continuing to build on this classic system. People have studied these positive effects, but then there's been sort of a separate field of studying all the negative things that animals can also transport. And so this was nice because it was bringing both within the same organisms in the same place. There are pretty few studies that have really brought that together, which I think is important when we think about this holistically.
But while this may be a start, there's more work to be done to fully understand the big picture. This work is based on combining data sets, and in some cases, data was lacking on specific contaminant levels in specific species in specific years, say. Lily says that a lot more data needs to be collected moving forward, particularly as things like climate change and regulatory changes could alter things.
I think that's a good next step going forward to start monitoring for these different good and bad compounds, because these two fields have been so separate. So you might have one fish that had its contaminant contents measured and another fish that had its fatty acid contents measured. But it would be nice to be routinely measuring these different things from the same individuals. I think would be strategically a good idea because of the importance of fish, not only for these ecosystems, but also for human communities as well.
Jess agrees that more effort to collect data moving forward will add further nuance to the work. She says that this study helps fill in the gaps in how chemicals are cycled and what shifts in the populations of different types of salmon could mean for this cycling in the future.
One of the biggest contributions of this work is that it puts numbers on the amounts of materials that salmon are moving at a spatial scale that we can compare to some of these other major movements of chemicals around Earth's systems. We can compare them to how much of this material is being moved by a river or how much is being moved around through atmospheric deposition. We need to understand where these materials travel to, where they end up, and that helps us understand where they might have their influence.
Jess Brandt there. You also heard from Lily Twining. To read Jess's research paper and Lily's News and Views article, look out for links in the show notes. It's Nobel's week this week, so as very much is tradition on the Nature podcast, Flora Graham, senior editor of the Nature Briefing, joins me to discuss this year's winner. Flora, how are you doing today?
I'm doing great. Always excited about Nobel week, especially to be on the podcast.
Excellent. Well, this is the 5th year you and I have done this, and we've grabbed a little space in Nature Towers to go through some of the winners this year. So I guess let's start at the start then on Monday, and the 2024 prize in physiology or medicine, and that was awarded to 2 geneticists who discovered microRNAs.
That's right. This is not to be confused with mRNA, which not only transformed the vaccine landscape, but also won the Nobel Prize last year. This is microRNA.
And these are tiny little bits of RNA that seem to have a multitude of different uses, from embryonic development to, cell biology, in some diseases as well. And the winners in this case are Victor Ambros at the University of Massachusetts Medical School and Gary Ruvkin from the Massachusetts General Hospital. Now, we often like to talk about the funny way someone found out, and I heard that when he was phoned up, he wasn't sure if it was a wind-up from one of his friends or not, but it was legit, and he is one of the winners for this year. And the two of them were postdocs in a group when they published this kind of first key discoveries on these microRNAs in 1993.
And And I think what's interesting is this discovery was not seen immediately as something that was going to change the game. For quite a long time, when this microRNA structure was found in our old favorite C. elegans, it was thought to be, a roundworm thing. Now we know that microRNAs are existing across the tree of life, and this field of research has absolutely exploded since then.
Yeah, and they kind of add an extra level of complexity to gene expression and regulation. Because obviously, I guess, high school level, we have the DNA. The DNA gets transcribed into mRNA, messenger RNA, and that then gets translated into proteins. But researchers have learned a lot since that kind of dogma was established. And there is this entire extra level of on-off switches and all the rest of it that goes on. And these microRNAs are one of these. They are tiny little molecules, but they have a really outsized role in which genes are switched on and off. But quite difficult to study because like a single microRNA can affect the expression of 100 odd genes. So I think they've been quite difficult to exactly pin down. But as you say, they were thought of as something of an oddity from the roundworm C. elegans. But yeah, they are across the tree of life.
And the thought is that microRNA was fundamental to the evolution of all kinds of types of life, because it really is what allows the same DNA blueprint to create all the kind of wonderful variety of cells that exist.
Yeah, absolutely. And they play a crucial role in development, physiology, potentially behavior, and all sorts of other traits as well. And of course, they are of great interest in the therapeutic sense. There are a few drugs in development, but getting them into cells has, as I understand, been a challenge. So although this research was done some time ago, we're still kind of at the infancy of where it might lead.
And I have to throw in one fun fact. I know a lot of our listeners are early career researchers or thinking of becoming them. And I did discover while reporting this story that Victor Ambros was actually denied tenure by Harvard University very soon after he published his seminal paper describing this work. So he, of course, went on to some success. So, let that be a ray of hope for us all. It's not too late for each of us to go on to win a Nobel Prize yet.
Well, you never know, right? We've been overlooked yet again, Flora. But I think also it's nice that this is kind of another highlight of the importance of curiosity, right? Like the way that these microRNAs were discovered was there were some worms in a lab that looked a bit funny. And from looking at these and unpicking how it works, say this whole field has opened up.
Yeah, I think that's a point that is often made by these winners, is that it really shows the value of fundamental research. And I know Gary Ruvkin really emphasized that this is a triumph for fundamental research and should be used to celebrate and encourage that kind of research.
Well, let's move on to Tuesday and the Nobel Prize in Physics, which was shared between two researchers, John Hopfield from Princeton University in the US and Jeffrey Hinton at the University of Toronto in Canada.
Woo! Shout out to Canada. Always happy to see a winner.
And their prize is shared for their development of tools to understand the neural networks that underpin the boom in artificial intelligence.
That's right. These models have ultimately proven to be the underpinning of these enormously successful artificial intelligence systems that we're seeing today.
And in 1982, then Hopfield, who is a theoretical biologist but with a physics background, came up with this idea of a network that describes the connections between virtual neurons as physical forces. And obviously, a lot of research has gone into this, but it ultimately became known as associative memory, which allows this network to, heavy inverted commas, recall things. And it's similar to a brain trying to remember a word or a concept based on related information. And Hinton kind of took that idea and built on it using principles from statistical physics. And his work involved building probabilities in and could be used to generate images based on examples of what the network was trained on, which obviously is quite central to some of the AI networks that we know about today.
I mean, I think what's really notable here is that the step change that this took from previous types of computational techniques was that it could learn from examples, it could learn from complex data. And this would have been quite difficult for conventional software that's built more on the step-by-step calculation.
And maybe while they're kind of idealized versions of brains, Hinton did write in Nature Neuroscience in 2000 that they are, quote, grossly idealized models that are as different from real biological neural networks as apples are from planets, end quotes. And yet they have proved to be incredibly useful and built upon, and here we are in the 21st century.
One thing to note is that Hinton has actually become a really vocal critic of artificial intelligence and how it's progressing. He has expressed a lot of admiration for the way that biologists have managed to put guardrails around things like cloning and slightly successfully keep the genie in the bottle in that sphere. Hinton actually quit his job at Google last year, specifically so he could speak freely about the dangers that he was predicting could arise from the application of AI technology. In fact, he's said, a part of him actually, from one perspective, regrets his life's work. And he's said that all he can say is, if he hadn't done it, someone else would have. And now he and other giants in the field are really standing up and saying it's up to us to ensure that this doesn't create many of the negative knock-on effects that a lot of people have already flagged.
Yeah, I think guardrails are something that he has talked about. But in terms of how this work has been used, it seems like it has ultimately had a bunch of applications, certainly in biology, and I think we'll talk about one of those in a minute, but also in physics as well.
That's right. I mean, I have to recognize that there's a lot of people saying, is this physics? This is the Nobel Prize for Physics. Okay, fair enough. They quote from the Nobel Awards Committee, they use the tools of physics to create these things. But really, is this physics? Well, I mean, Hopfield actually addressed that question himself back in 2014. He actually started as an extremely successful condensed matter physicist and was really award-winning in that field, and kind of went looking around saying, I need a new challenge. And that's how he ended up moving into this area. Hopfield's parents were both actually physicists. And so he said, to me, growing up with a father and mother who were both physicists, physics was not subject matter. The atom, the troposphere, the nucleus, a piece of glass, the washing machine, my bicycle, these were all incidentally the subject matter. But the central idea was that the world is understandable and physics is a point of view. So that's one really nice way of looking at it. And Hopfield would certainly argue that the work he's doing is physics in that sense.
Well, certainly, I mean, his work was motivated to, I think, understand certain phases of matter. But I think what's kind of an interesting full circle is that these models are being used in neuroscience to further understand the brain, because of course there's a huge amount of information coming from individual cells that researchers are recording. So using these models to try and parcel that out and work out what's going on, say it was inspired by the brain and it's being used to understand more about the brain as well.
Oh, absolutely. I mean, talk about inputting complex data sets that typical computational methods would have difficulty with. I think the functioning of the brain is probably an ideal example of that.
Well, I think I've kind of foreshadowed today's prize then, which is the Chemistry Nobel, announced only a few hours ago as we're recording this. And neural networks do seem to be kind of central to this prize, and it's shared by 3 winners.
That's right. It's split in half. One half has gone to 2 researchers at DeepMind, which is the artificial intelligence company that's owned by Google, Demis Hassabis, who actually co-founded DeepMind, and the theoretical chemist John Jumper. And they won their half of the prize for leading the development of AlphaFold, which is an AI tool, and it has recently and very swiftly transformed biology. I mean, often with Nobel Prizes, you hear this was for research that was done decades ago. But in this case, we're talking about the last couple of years where AlphaFold has really come to the forefront and transformed its field of protein structure prediction. The other half of the award goes to biophysicist David Baker. He actually led the design of another influential protein tool, which is called Rosetta Fold. But he won primarily for his work not just predicting the structure of proteins that already exist, but dreaming up new ones.
And obviously, when we say protein folding, I think it's important to maybe explain what that means. So a protein is essentially beads on a string of amino acids, and these will fold up due to various chemical and energetic factors. And working out what that 3D structure is really hard to do, because these beads, these amino acid strings, can be really, really long. And so by knowing the shapes, you get a sense of how they work, how they interact with other things, and maybe how they can be tweaked to do new stuff. So it's a big field in basic research and also in medical science and all the rest of it. And with that being said, then let's talk about the work the winners won their prizes for. Let's start with the DeepMind project AlphaFold then. Now, a lot of things are thrown about AlphaFold, game changer, all this kind of thing, but it does seem that biologists talk about the before AlphaFold era and the after. And this work predicts protein structures using a neural network, there it is, that incorporates similar structures from databases of hundreds of thousands of structures and millions of sequences from related proteins. And as you say, it hasn't been about that long. It debuted in 2018 and it won a protein structure prediction prize. But the second version, AlphaFold 2, this seems to be where everyone started taking notice because it was so accurate that its results were indistinguishable from experimentally solved protein structures. And the team have gone on to develop AlphaFold 3 as well, which incorporates other things that attach to proteins, drugs and what have you. But it is still in its early days, right? And its impact on science, I guess, isn't entirely clear yet.
I think that one of the key things here is that this did work that would have taken years in effectively minutes. So the team already in 2021 announced that they used AlphaFold to predict the structure of basically every protein made by humans, as well as a bunch of proteins from other organisms and things. So when you have that kind of power, even though there are certainly limitations to this tool, it just accelerates all of the things that maybe, not that long ago thought to be impossible, really being able to predict the structure of proteins from knowing their amino acid sequences, which really allows people to start thinking about new problems that they want to solve.
And DeepMind will get the headlines, I think it's fair to say. But David Baker and his colleagues developed a different system then, and it too was one of the top entries to the protein folding competition. But say now it's really been prolific at, say, using machine learning to think outside the box, I guess. Think about new stuff that doesn't exist in nature.
Yeah, Baker and his research group have been working on this idea for quite a long time. It was back in 2003 that Baker's team created the first protein with an entirely novel structure. And now they're using these tools to design proteins that do useful things. For example, catalyze a particular chemical reaction. So they specify the amino acids responsible, and then they let the AI, what we call hallucinate. It's quite good at coming up with very convincing protein structures, and not all of those will exist already in nature, but what they found by testing them actually in the lab, by getting bacteria to actually produce these, they found that some of them do have the effects that they're looking for.
And I think it's interesting that with tools like this and, with AI more generally, there are worries that it will replace humans, replace researchers. But it seems like with these tools, the opposite has happened to an extent. These aren't replacements for experimental studies, but they are an accelerator. They are this jumping off point that allows researchers to really ask more advanced questions.
Yeah, it does open the door to new kinds of research, because let's remember that it makes predictions, but it has limitations. It's not necessarily equipped to predict complex interactions of proteins. It's not what it was trained to do. It's not necessarily equipped to predict mutations, for example. So it's not as simple as, a protein creating replicator or something like that. It's a tool, but everything still needs to really be tested experimentally.
Well, those are the winners this year then, Flora, and congratulations to them. But of course, we seem to talk about it every year. There wasn't a lot of diversity in this year's winners. They were all men.
It's a bit of the elephant in the room, I'd say, when the prize is announced. I must admit, we all kind of look at each other and think, oh, this again, because every year we discuss, what the Nobel Committee has agreed to do to try to better reflect the makeup of scientists. And it does feel a bit disappointing this year that we haven't seen the results of any of those efforts really come to fruition. I do think it's worth mentioning that the Nobel Prize winners, almost without exception, really do stand up and say, even though the prize can only be given to a small handful of people, that does not reflect the fact that these enormous successes could not have been achieved without their fellow researchers, their students, and they often stand up and say, this isn't just for me, this is for all the people who did this research with me.
Well, Flora, thank you so much for joining me. Shall I set a clock for 12 months' time? Should we do it again?
Can't wait.
Flora Graham, thank you very much.
Thank you.
Nature's Flora Graham there. For more on Nature's coverage of the Nobels, head over to the show notes for some links.
And that's all for this week. We'll be back again next week with more news from the world of science. In the meantime, you can keep in touch with us on X. We're at Nature Podcast, or you can send an email to podcast@nature.com. I'm Nick Petrichow.
And I'm Benjamin Thompson. Thanks for listening.